record and share the screen. Do you see energy and enzymes?
Yes, sir.
Right, then I will make it big. The sun, as far as biology is concerned, the sun is the only source of energy. There are other sources of energy on the planet, but living things benefit from the sun. So we get energy from the sun in the form of light, and somehow that gets converted into chemical energy. And that somehow means through photosynthesis. We'll talk a lot about photosynthesis later. So plants are the producers. Chlorophyll is the most abundant molecule on the planet, biological molecule on the planet. And it's because chlorophyll and photosynthesis are the source of all energy for living things. So the plants are producers. Non-plants, non-green things are the consumers, and the nutrients between producers and consumers are constantly recycling. So what is energy? It's the capacity to do work. What is work? Work is the ability to move matter in a direction that it would not move if left alone. Energy is measured in calories. So 1 calorie, definition of a calorie, is the amount of energy required to raise the temperature of 1 gram of water 1 degree centigrade at sea level. When we talk about calories, we're usually not referring to real calories. We're talking about the diet, and diets are measured in kilocalories. So a dietary calorie is 1,000 calories. So if you're on a 2,000 calorie a day diet, you're really talking about 2,000 kilocalories or 2 million calories. Okay, what are the forms of energy? Potential and kinetic. So potential energy is stored. For example, a rock sitting on a hill is potential energy, or ions on one side of a membrane. They're not moving, they're sitting there. That's potential. Kinetic energy is when the rock tumbles down the hill, or water flowing over a water wheel is kinetic energy. And if the ions on one side of a membrane start to move through the membrane, that goes from potential to kinetic energy. So potential energy and kinetic energy are constantly being interconverted. And that brings us to the laws of thermodynamics, the first law and the second law. They're both relevant to biological systems. So the first law is that in a closed system, energy can be neither created nor destroyed. It can change forms. So the amount of energy present at the beginning of the universe will be the same at the end of the universe. The form of energy at the end of the universe will be different. So that raises the question, how will the universe end? Of course, we don't know, but astrophysicists do have hypotheses. One is that the universe will expand forever. And the reason for thinking that is that right now, the universe is expanding. In other words, everything in the universe is moving away from everything else in the universe. and it doesn't seem to be showing any signs of ever stopping this process. It keeps going on, and in fact is speeding up. So if that happens, and all stars will burn up their fuel eventually, leaving only black holes. Another hypothesis is the opposite of that, and that is that the universe will stop expanding at some point and start to collapse. And it'll continue to collapse backward. In other words, sort of a backward Big Bang, so that in the end, all energy will end up in an infinitely small spot. And then there's a hypothesis based on dark energy, which is the opposite of gravity. And it's similar to number one in that dark energy causes things to pull apart from each other, from atoms to planets, galaxies, and the universe itself has the problem, the issue of dark energy. So we don't know how the universe will end. None of us will be around to see it, but it's interesting to think about, speculate. Then the second law of thermodynamics, energy transformations result in increased disorder, and that's called entropy. So some of the energy is no longer available to do work. And the term entropy is a measure of the amount of disorder in a system. Now that becomes relevant to biological systems because the implication of the second law is that since disorder is always increasing, to keep a system orderly requires a constant input of energy. Without that, all chemical reactions go to equilibrium. And since a biological entity is nothing but a collection of chemical reactions without a constant input of energy, all those chemical reactions go to equilibrium and the organism is dead. In other words, if there's no energy going in and no energy coming out, the organism is dead. So the point is, life requires constant input of energy. Now, why do cells need energy to survive? Well, I just said why, because they die if they don't. They're subject to becoming disordered because of the second law of thermodynamics. They remain alive and healthy if they have a constant input of energy. And that brings us to electromagnetic radiation. It's a form of kinetic energy. It's energy that's active. It consists of a spectrum of electromagnetic waves that differ in wavelength and energy content. This is something that's always hard for me to conceive, that waves and particles are the same thing. A wave can also have the property of a particle, and when it does, something for you to remember, when a wave behaves like a particle, it's called a photon. A photon is a quantum of light. It has no mass. It carries energy, energy proportional to its radiation frequency, but it has no mass. Well, that's a photon. Well, here's the electromagnetic spectrum. And the white part here, the narrow band of white, is visible light. That's what we can see with our eyes. At the top, you can see that it goes from low energy on the right to high energy on the left, and from long wavelength on the right to short wavelength on the left. And the squiggly line here, it represents that you have long wavelengths at the right-hand side of the spectrum and very short wavelengths at the left-hand side. Now, what are these? Well, starting at the right, there are radio waves, microwaves, infrared, then there's visible light that we can see, then UV, X-rays, and gamma rays. Now this visible light that we can see ranges from red to yellow to green to blue. And this becomes important when we talk about plants and photosynthesis, because why are plants green? It's because they absorb all the wavelengths of light except green, and they reflect green. So when plants take in energy from the sun, they're absorbing all the wavelengths except for green, which they reflect. Now, I thought you would be interested in knowing the wavelengths of radiation that we can't see. What are they and what do they do? What are they good for? Well, starting with the most energetic, gamma rays, they're produced by the hottest objects in the universe, such as neutron stars and regions near black holes. On Earth, they can be generated by nuclear explosions, by lightning, and by radioactive decay, which is probably the most useful to humans of possibilities for gamma rays, because they can be used in cancer therapy to kill cancer cells and not healthy cells. They can be used to sterilize equipment and to be used in tracers for doing research. X-rays. You all know about X-rays. They're a penetrating form of radiation, pass through living things and leave shadows of the things inside living organisms like bones and organs. So they're used in medicine. When you go to the airport, go through a scanner at the airport, that's X-rays. UV radiation is found in sunlight. It's used for skin tanning, convert sunlight to vitamin D. It's also used for sterilizing equipment and for coronavirus eradication. So in large public places that wish to be cleared of coronavirus, they're usually exposed to UV light for some period of time. And then we have the visible light, which you've already talked about. What's infrared? Well, it's invisible wavelengths, longer than visible red light, and shorter than microwaves. And it's used for wireless communication. So for example, a garage door opener or your TV remote is probably using infrared. It's used for night vision goggles and interestingly, heat-seeking missiles. Microwaves, again, this is something that's part of everybody's everyday life, microwave ovens. It's a portion of the spectrum with wavelengths between a millimeter and a meter. So these are distances that we can easily imagine in the real world, millimeter being very short, a meter being about 3 feet. It's non-ionizing, so therefore it's relatively safe. And the way it heats food is by causing molecules of fat and water to vibrate. And finally, radio waves are the longest. They can be anywhere from a millimeter to 100 kilometers. 100 kilometers is about 60 miles. So that is a very long wave. And these waves are used for long range transmission of radio and TV signals. Now, getting back to the electromagnetic spectrum, how can we capture and make use of the energy that's available there? Photosynthesis. It's a process that's performed by plants. It captures the energy in the part of the electromagnetic spectrum that we see as visible light. And as I said, part of the visible light, the visible spectrum is green light. And the reason plants are green is because they reflect green light and absorb the other wavelengths and use that, convert that to energy, to chemical energy. Well, chemical reactions, cellular metabolism is the sum of all the reactions going on in a cell at any given point in time. Thousands of individual reactions. And every reaction in a biological system is mediated by an enzyme. That's why this section of the course is called Energy and Enzymes. So enzymes are biological catalysts. They speed up reactions in a cell without themselves being changed, not permanently changed. They continually recycle. So without enzymes, reactions would be far too slow for biology. Because what enzymes do is reduce the amount of energy that it takes for a reaction to occur. So this is the way enzymes work in sequence. You have compounds A and B. Enzyme 1 can convert them to C. Enzyme 2 can break them apart into D and E, which are different from A and B. And enzyme 3 can convert them to yet another compound, F. So there are two types of chemical reactions in a cell, endergonic and exergonic. So endergonic require an input of energy. Think of it as running uphill. Exergonic reactions release energy. Think of it as going downhill. So that's illustrated here in this diagram on the right, where carbon dioxide and water are being converted by an energy input in an endergonic reaction to glucose and oxygen. So carbon dioxide and water go to glucose and oxygen with an energy input. Now, if you break down glucose in the presence of oxygen, in other words, oxidize it, energy comes out. And that is the energy that living systems use to keep themselves alive. So here you can see, you can visualize what enzymes do and familiarize with the term activation energy. So in the top diagram, man's pushing the rock up a hill and the hill is very high. And so if this were a chemical reaction, converting sucrose to its component parts, glucose and fructose, without an enzyme, it takes a lot of energy to do that. If you add an enzyme in the bottom diagram, the enzyme being sucrase, the enzyme will bind to sucrose, break it apart into glucose and fructose, then release it without itself changing. And so now the activation energy, you can see, is very small compared with the activation energy without the enzyme. The net energy released is the same in both cases. So how do enzymes work? Well, they orient the substrate. They induce strain in the substrate. And as I'll show you in a minute, the strain usually has the effect of breaking bonds within the substrate. and they can temporarily add chemical groups to the substrate. Again, you can see the process of an enzymatic reaction, energy plotted against time. And again, in this case, converting glucose and carbon dioxide, or no, phosphate, a phosphate group, the glucose 6-phosphate, the activation energy is reduced in the presence of an enzyme. Now, what are the ways that an enzyme can actually operate? So the blue object is the enzyme. The little other colored objects are substrates, potential substrates. So you can see in the center diagram, 2 of them have bound in the active sites of the enzyme. And the enzyme performs an action. In this case, it brings the 2 products, the 2 substrates together to make 1 product. So the points here, you have enzyme, you have substrate, and you have active site. And it's often equated, enzymes and substrates are equated to lock and key analogy. The shapes of the enzymes and the substrates are critical. That's how they recognize each other, by shape. So each enzyme can react with only 1 kind of substance. They're very specific, and the reason they're specific is because of the exactness of the shape. The enzyme shape determines which substrate will fit. So if you visualize a lock and a key, the enzyme is the lock, the substrate is the key, and if it fits, the reaction goes forward. On the right here, you can see an example where when the key fits, a correct fit will react. If the key doesn't fit, there's no reaction. In other words, if the substrate doesn't fit into the active site of the enzyme, nothing happens. Now this is something that you really need to remember, get on your mind, because it indicates how an enzyme actually works, how it actually converts a substrate to a product. So at the top here, you can see substrate in green, the enzyme in yellow, and the active site. So number 1, and if you look to the right, number 1, the substrate binds to the enzyme active site. So the active site here is indicated by the arrow. Once it's in the active site, 2 and 3 happen. So 1, the enzyme changes shape. And as a result of that, the bonds in the substrate are either broken or weakened. So number 4, if the bonds are broken, then when the enzyme releases the substrate, it's now 2 products. Products are released with little expenditure of energy. The only energy needed is whatever it takes for the enzyme to change shape. And at the bottom, you see the same thing illustrated in a different way, with the enzyme, the substrate fitting into the enzyme active site, the enzyme changing shape so that it changes the substrate into a product. Okay, everybody get that? The way an enzyme can work, it's not always the case, but the common way for an enzyme to work is to simply change shape and weaken the bonds. And you remember what the bonds are. If the substrate is a protein, there are 4 different kinds of bonds holding a protein together, and those bonds are, most of them are quite weak. And so they're relatively easily broken if an enzyme changes shape. So this illustrates how an enzyme gets involved in a reaction but does not itself change. If you start with number 1 here, the enzyme and the substrate are available. The enzyme in this case again is sucrase, has an active site, and the substrate is sucrose, which is a molecule composed of glucose and fructose. So that substrate fits into the active site. Water is added, and out come glucose and fructose as individual molecules. The enzyme hasn't changed. It goes back to being a free molecule with an active site. Substrate rate effects. So we have the concentration of substrate plotted against the reaction rate. That means the speed with which the enzyme is converting substrate to product. So without an enzyme, the reaction time is slow. It takes a long time to convert. As the concentration of substrate increases, the reaction rate increases, but it's very slow. Whereas with an enzyme, it reaches the maximum rate fairly quickly with a much lower concentration of substrate. Enzyme denaturation. Enzymes, being proteins, can be damaged permanently by exposing them to extreme environments. So if the temperature is too high, they'll denature. The bonds, the tertiary structure of the protein is destroyed. Or if the pH is either too high or too low, or if the salt concentration is too high or too low. So most proteins, once they're denatured, cannot renature again. They're cooked, they're done. So the enzyme's tertiary structure is destroyed, cannot function anymore. So this shows you some examples of how enzymes are adapted to the environment in which they function. So the top panel shows the optimum temperature for a typical enzyme in the human body. The human body normal temperature is about 37 degrees centigrade. And if you look at the x-axis here, temperature concentration increasing, you can see that the optimum, in other words, the temperature at which the enzyme, most human enzymes run the fastest, is at about 37 degrees. On the other hand, to the right, the optimal temperature for an enzyme from a thermophilic bacterium is very high. In this case, about 75 degrees. And you remember when we talked about PCR, polymerase chain reaction, it's absolutely dependent on an enzyme called DNA Taq polymerase, which was isolated from a bacterium in a thermal vent, and it can actually tolerate temperatures as high as 95 degrees without denaturing. So most enzymes would denature at a temperature far below 95. Taq polymerase can tolerate it. It's not its optimum for function, but it doesn't denature. However, for optimum function, Taq polymerase has an optimum about 75 degrees. And that you can see here. Another environmental condition that enzymes are adapted to is pH. So in our stomachs, the pH is about 2, very low, very acidic. Pepsin is an enzyme that starts digesting food as soon as it enters the stomach, and it has a pH optimum of 2. On the other hand, when you remember when we talked about cystic fibrosis and the pancreas, the pancreas produces digestive enzymes, one of which is trypsin. The pH of the duodenum, where trypsin goes to function, is between 6 and 7, and the pH optimum for trypsin is about 8. So enzymes are adapted to function optimally in the environment where they have to work. Make sense? Anybody not understand it? We'll have a worksheet next time. We can talk about these things further. So enzymes usually, as proteins, usually don't act alone. So what are their partners? Well, they are categorized as cofactors or coenzymes or prosthetic groups. Well, what is a cofactor? It's an inorganic component, often a metal, that's required for enzyme function. Examples of cofactors are iron. For example, hemoglobin needs 1 atom of iron for every molecule of hemoglobin in order to carry oxygen. Other metal cofactors are copper and zinc. Coenzymes are very often vitamins. They often combine with an apoenzyme protein to form the active enzyme. And the coenzymes, in contrast to cofactors, are usually organic substances, as are prosthetic groups. Prosthetic groups are a non-protein part of an enzyme. For example, heme, again, heme and iron together in hemoglobin is what makes hemoglobin capable of carrying oxygen. So if a prosthetic group is a non-protein part of an enzyme, what is the protein part? What is the protein made of? Not all of these things on the table. It's too easy. Protein is made of amino acids. ATP. You've probably all heard the term ATP is the energy currency of the cell. It's adenosine triphosphate. It's the main energy carrying molecule in cells. So this goes back to, remember when we talked about macromolecules, DNA. Adenine is one of the 4 bases that goes into DNA and RNA. But in this case, it's not part of a genetic molecule, but rather an energy carrying molecule. So this breaks it down, the structure down into its various components. Adenosine is adenine plus ribose, or in the case of DNA, deoxyribose. AMP is adenosine monophosphate, so it's adenosine with 1 phosphate group. ADP, diphosphate, has 2 phosphate groups. And adenosine triphosphate, ATP, has 3. And it's this last phosphate group that's attached to the one adjacent to it by a high energy bond. So when that bond is broken, energy is released and can be used for doing work. There are other molecules that can release energy, usually from a phosphate group, but ATP is by far and away the most commonly used in biological systems. This is a space-filling model, the top left here, where the adenine is represented in blue, the ribose in the middle, and then the 3 phosphate groups are the orange balls on the left-hand end. So ATP and ADP are interconvertible, and they go back and forth constantly in a biological system. So what you see here in the diagram, ATP at the top with the 3 phosphate groups, and ADP at the bottom with 2 phosphate groups, one having been cleaved off to release energy. So to make ATP is an endergonic reaction. Energy goes in order to join ADP and a phosphate group to make ATP. When ATP is hydrolyzed to ADP, energy comes out. It's an exergonic reaction, and you end up with ADP phosphate plus energy. And this interconversion between ADP and ATP is going on constantly, and the only reason that it can go on is there is a constant input of energy to take the expended ADP and convert it back to ATP. There are 2 kinds of metabolic pathways, anabolic and catabolic. I like to think of them as anabolic pathways build up and catabolic pathways tear down. So anabolic pathways build up. They link together simple molecules to form complex molecules. The anabolic reactions store energy in chemical bonds. And almost always anabolic pathways are endergonic. They're not spontaneous. They have to have a free energy input. So that's anabolic. Catabolic means tear down, break down complex molecules to form simple ones. And in the breaking of the bonds in these complex molecules releases energy. And so the catabolic pathways are exergonic. They have a spontaneous release of free energy, which is then used by the cell to do work. And I have here some attempts to illustrate anabolic and catabolic. So the simplest way to look at it is at the top. An anabolic pathway, you see there are 4 small molecules. Those 4 small molecules plus energy will make a complex molecule, a polymer, with energy stored in the bonds that join the individual subunits. So energy is stored in those bonds. And about metabolic pathway. Now, to get energy out of them, we have catabolic pathways in which large molecules, almost always polymers, are broken down into smaller ones. And as that happens, as the bonds here break, energy is released. At the bottom, I tried to illustrate catabolic and anabolic as really pathways going in opposite directions. So if you look at the left, catabolic, stage 1, you have the polymers. Proteins are polymers of amino acids, polysaccharides, polymers of sugars, and lipids, polymers of hydrocarbons. And these polymers are broken down into their various subunits, which are then converted to a common carrier called acetyl-CoA. And then in a series of reactions called the citric acid cycle, or the tricarboxylic acid cycle, we'll talk about that later, ATP is released. So energy is released. You start out with these polymers, and any one of them or all of them can be broken down and energy can be extracted at the end of their reaction steps. And on the right, there's both anabolic and catabolic. I wanted to emphasize that these reaction pathways can go backward, in which case it's an anabolic pathway. So you can start with the citric acid cycle, which is this blue ball, and it can go backward to either fats, proteins, or carbohydrates. Glycogen is a carbohydrate. So any questions about catabolic and anabolic? You have a half hour to spend on your quiz today, so I shouldn't, I intend to stop this, this lecture at 45 minutes, which it is right now. I think I'll stop here and let you go, and I'll finish this slide set on Monday, along with a worksheet, at least one worksheet, maybe two, which we'll discuss, and discuss is underlined. I'd like to have a true discussion of all of the things in energy and catalysis with everyone in the class participating. So any questions about today or what we're going to do on Monday? No questions? Okay. Then I'm going to stop sharing the screen. And if there's nothing else, I'll let you go and you can study for the quiz or take the quiz. Just get it in before midnight. And really a silent bunch today.
Thank you, Professor.
Okay, you're welcome. Glad you came to class. And I will see you on Monday.
Have a good day.
I'll have your quizzes graded in the meantime. Okay, bye.